And then we can go on with the next speaker. It is Suzanne Vernon from Bateman Horne Center, a center in Utah which has been very active in the research of ME/CFS. And she will talk about one of the important phenomena we see in ME/CFS, namely cognitive impairment. And they have done studies in order to try why there are cognitive impairments in patients with long COVID and with ME. So please, Susan.
Now on? Yes. I'm Suzanne from the Bateman Horne Center. The Bateman Horne Center is located in Salt Lake City, Utah. I actually live in Colorado and have, I visit Salt Lake City quite often, quarterly, to conduct clinical research. And we have made it our mission at the Bateman Horne Center to use the Institute of Medicine criteria, which is a clinical diagnostic criteria, not to manage our ME patients, and also to operationalize the criteria so that we can use each one to potentially as outcome measures for clinical trials. So 2 arms, clinical management as well as clinical research. So that's what I will focus on today. Just wanted to, here's my few disclosures. We have funding from NIH. Some of the results that I will show you today come from a study that was funded by Terra Biologicals for a nutraceutical, medical food, and all of these results do not necessarily represent the official opinions of the NIH and other funders. Again, at the Bateman Horne Center, our model is one of really learning from our patient population. So we bring patients in, we listen, we learn, we manage, we treat. We use that information then to design our clinical research studies, our clinical trials. We also use that information for education. And if you go to our website, you will see there are a number of educational resources, both for patients and for healthcare providers. They've been very well received throughout the world. And again, here's the roadmap that, and the reason that we've used this as a roadmap, Dr. Lucinda Bateman, the founder of the Bateman Horne Center, was one of the 18 or so members of the committee on the Institute of Medicine committee that came up with this evidence-based clinical diagnostic criteria for ME/CFS. When this came about, it was probably one of the first easily operationalized diagnostic algorithms for ME/CFS, and it included, most importantly, post-exertional malaise. So as you can see, if the patient presents with profound disease and there's a substantial decrease in function, not just fatigue, a substantial decrease in function that has persisted for at least 6 months and is associated with fatigue, profound fatigue, and also includes post-exertional malaise and unrefreshing sleep, there is a very good chance that ME/CFS is the cause of this. You also are required to have either cognitive impairment or orthostatic intolerance, or both. All of these symptoms have to be present frequently, at least half of the time, and also have to be moderate to severe in their intensity. Again, this is what we use as our roadmap, our guideline, and it works very well. And when the pandemic hit, we knew, because together I'm dating myself here. Together, Cindy, Dr. Bateman, and myself have more than 60 years of clinical research and medical experience at ME/CFS. We knew with the pandemic there was going to be some post-COVID ME/CFS. So what we have intentionally done is applied this criteria not only to our ME patient population, but also our long COVID population. Today, I'm going to focus on two aspects of that criteria. Specifically, how we use objective and passive assessments to assess impaired function, physical function, okay? Mostly in the context of clinical trials, making this so that we can use it as impaired physical function as a primary outcome measure for clinical trials. And then I'll focus on objective assessment of cognitive impairment before and after orthostatic challenge. Again, as a way to get very objective measures of cognitive impairment for clinical trial outcome measures. This has been one of the major barriers for the pharmaceutical industry to get interested in ME and likely long COVID, because symptom surveys, symptom assessment questionnaires are good, but they're soft measures. And for pharmaceutical companies to get interested to apply their meds, they really want to have solid, objective measures that aren't necessarily subjected to subjectivity of the patient. So here we go. When I started working with Cindy several years ago, I learned something right away, that in the clinic, she uses a very simple questionnaire that she called HUA, that stands for Hours of Upright Activity, to assess how severely ill the patient is. And it's over 24 hours. How much time do you have your feet on the floor standing up? Do you have your feet on the floor sitting down? Do you have your feet out and then you're lying down? And so patients would respond. And by that response, she was able to tell how ill they were and also monitor them over time. In a publication, we were able to show that response to HUA also correlated with orthostatic intolerance symptoms. So I said, we have to be able to figure out, to use some type of technology to be able to collect that information in an objective way and also passively. Because over the years, what we've heard from patients is, I'm so tired of answering questionnaires. So many questionnaires that I have to answer through research. And also, it's the problem of recall. I don't really know how much time I have my feet on the floor, or you have to think about it. And it, again, becomes very objective. So given the technology that we had, I said we have to be able to do this objectively. And importantly, I also wanted to do passively, where you didn't have to think about it. You're wearing a ring or you're wearing a watch. You don't have to think about it. And I wanted the data to be collected all the time. So this is a little device. This is a clinical research device. This is not a Fitbit or anything. It has all the same sensors in there, but we used it because we could access the insides of the sensor to make it do the things we needed to do. And we also, as you see in this picture, it's worn on the ankle. That will become clear in the next couple of slides. So we did a choreographed study. Why is that important? So this is so cool. I mean, this was all new to me. I worked with a mechanical engineer at the University of Utah, Shad Roundy. And what we did was we put 5 volunteers into a sensor lab, a motion sensor lab. So motion sensor labs, you have sensors put all over your body, and we had them do all types of positions so that we could determine when exactly are you upright and when are you not upright. And to show you, I mean, this just illustrates how incredibly accurate this type of assessment is. That's Gollum. This is Gollum right here. What we were able to determine is that it is essential to have the device somewhere below the knee. And if you think of it as a cone below your knee, it's a 39-degree angle all the way around. So anywhere outside of that 39-degree angle is not upright. Anywhere inside is upright. Very easy. Theoretically, you could wear a Fitbit on your ankle if you were able to access the insides of the Fitbit to manipulate, to access the data. What we were able to do with that is show, we studied 10 severely ill, 10 moderately ill, and 10 healthy subjects. And they wore the device, which we call uptime, for lack of a more clever term yet. What we were able to show is that severely ill patients spend on average have 0 to 20% uptime. Now uptime, the scale is actually time. The frequency is very, 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 very high, so we could actually get this down to seconds. But we'll think of it as in minutes. And then moderately ill have 20 to 30% of their time in an upright position, and then the controls have 50% or more in upright position. And that's shown not only in this scale, but also over here. And I think importantly about this slide is the confidence intervals don't overlap. And that becomes very important when you're thinking about outcome measures and determining the effect of a treatment. This is the data that has not been published yet, but this is we used uptime, the device, in a 3-month-long treatment trial of a medical food. And the primary outcome measure was fatigue, and we used uptime as an exploratory secondary outcome. So responders in this case are people who had significantly decreased fatigue. And what I did was looked at the enhanced responders, blue being the treatment arm, red being the placebo arm. You see that there were even some responders in the placebo arm. We've heard a lot about, why that might be. But there was a significant increase in uptime in the enhanced responders. This equates to about 45 minutes over a 24-hour period. Now, you might say, well, that's only 45 minutes. But 45 minutes when it comes to being able to be upright, to make a salad, or to take a shower, or to take a short walk, is, in fact, very important. So it was exciting to see that we could actually get a very objective and quantitative outcome assessment from a treatment. We've had people say, well, why don't you just measure steps and activity? So feet on the floor is this. Feet on the floor is sitting on a chair like this. My feet are on the floor. I'm still upright. STEPS misses all of that. STEPS does not get that. So STEPS is not a very accurate indication of upright activity. And that's also shown here. So we have ME/CFS, we have long COVID, we have healthy controls, and the confidence intervals overlap on all of them. Yes, ME/CFS has fewer steps, but it's not necessarily all that dramatically different or significantly different from long COVID or healthy controls. Whereas uptime, again, we do not have confidence interval overlaps, and we're able to distinguish between groups. So it's a very accurate, going back to our Gollum picture, it's a very accurate assessment. So I want to switch now to cognition. Again, uptime related to circulation, cognition, as we've heard, related to circulation or perfusion of the brain. One of the studies that really inspired us to think about how to objectively assess cognition in clinical trials was this particular study led by Dane Cook, where they assessed using a PASC test pre and post exercise. And the take-home message was from this was slow information processing through imaging. This was a very elegant imaging study that was done. And slow information processing is the most common, this was also cited in the IOM report, deficit for cognition. Other abnormalities include decreased working memory and attention. But it was this information processing, which is really how quickly can you make a decision, can you have an action, that we decided to focus on for cognition. And of course, again, this is related, we believe, that brain fog, cognitive impairment is related to orthostatic intolerance. Probably because of the cerebral hyperperfusion, the blood's not flowing above your belly button, so you are not getting the necessary energy and oxygen that your blood needs or that your brain needs to focus. Okay, so actually, where's Trudy? Trudy asked me this question yesterday. So yes, cognitive testing is really cool, but you know, I could be sitting here and be perfectly great. I get a great test when you give me this test. And I said, you're absolutely right. And this is why we integrated cognitive testing into an orthostatic test. How many of you are familiar with the NASA lean test? Excellent, quite a few. We use this, again, clinically for clinical diagnostic management, as well as in our clinical research and clinical trials. It's a very, very sensitive assessment of orthostatic intolerance. And for those of you who don't know what it is, it's a passive lean test, which means, as you can see, he's leaning against the wall. His feet are slightly about 6 inches away from the wall. His leg muscles are relaxed, so they're not engaged, so they're not influencing the blood flow above his belly button. They have a pulse ox, they're wearing a pulse ox, and blood pressure is taken every minute for 10 minutes. And at first, they're sitting down. We take a sitting blood pressure, then they stand up, and the test begins, and then they sit down again. So during those two sitting down periods, we have them do a cognitive test, which is called Dana Brain Vital. And I think I saw that David Systrom is using that in the trial. Great. So let's see. So again, before the orthostatic challenge, people take a test, a cognitive test, Dana Brain Vital, and then after. before I get to those results, I think we saw a similar symptom slide. This is what's happening with all of these symptoms before and after the 10-minute lean test. Blue, all the blue bars are ME/CFS, the red bars are long COVID, the green are healthy controls. Green, you can see, is all the way down here at the bottom. There's a little bit of symptoms, but not much. And I also will have to say that we have just as many healthy controls that take this test that faint sometime during it as we do ME patients or long COVID patients. Interestingly, the difference is that the healthy controls do not experience the same types of symptoms. This is what happens to the heart rate. Here's sitting down. Of course, as soon as they stand up, it goes up. And then as you can see, the controls have the lowest increase in heart rate, and it stays relatively steady. We have two ME/CFS groups here. This was important because there have been some reports by Ian Lipkin at Columbia that showed that there were differences in ME patients who had a shorter duration illness. Okay, so we studied ME patients who have been sick for less than 4 years. In the green line, ME patients sick greater than 10 years. In the blue line, long COVID in the purple. The long COVID and the shorter duration ME patients seem to track together with heart rate. This is blood pressure. Actually, this is a graph of narrowing of blood pressure. And narrowing of blood pressure is systolic pressure minus diastolic pressure divided by systolic pressure. And it's important to know that this type of narrow, extremely narrowed blood pressure is what is seen in anaphylaxis, septic, shock. Look what happens to the short-term and the long COVID patients within 4 minutes of the lean test. The blood pressure is just extremely narrow. Many of those subjects also were not able to complete the test. And interestingly, the longer duration ME/CFS patients, because they have adapted, because they have been treated over time, are quite similar to the control group. So that gives you a picture of what's happening from a hemodynamic perspective during this orthostatic challenge. Now, what's happening to cognition? This is the DANA Brain Vital. DANA stands for Defense Automated Neuroassessment Battery. It was a test that was actually developed for the Department of Defense in the US for soldiers that would have head injury in the battlefield, something that could assess traumatic brain injury right on the spot. It's a smartphone app, and it takes 5 minutes to do. This measures reaction time, pure and simple reaction time. 3 different types of tests. A simple reaction time, which is literally just touching, how fast you can touch the target every time it appears on your phone. This is procedural reaction time, adding a little bit more complexity in. A number will come up on the screen, and you have to touch which number it is. And then a little bit more complexity is a go, no go. So these last 2 adding in a bit of decision making to your reaction time. This is you gotta touch it when the bad alien shows, and a bad alien, that's a good alien because he's green, and the bad alien is gray. So slowed reaction time is one of the most sensitive measures to impaired cognition. And here's what we see. Kind of a busy slide, but these are the 3 tests. So simple reaction time, that's the target. This is the numbers. This is the alien. And then we have the red line in each one being our controls, and then the ME/CFS being green and blue, and the long COVID being purple. This is before, when they're sitting down. Number 1 is when they're sitting down before they take the orthostatic challenge, the NASA lean test. 2 is immediately after. 3 is 7 days later. And then 4 is 14 days later. Remember, this is an app on their phone. Everybody has it on their phone. They can do it from home. We get them all set up. It's perfect. And then we get all the data from through the portal. So what you see in each case for the patient group, not in the healthy control, is a dramatic decrease in reaction time immediately following the orthostatic challenge. And then in most cases, they go back up to their baseline level a little bit. It's basically to baseline level. What you should also recognize is this striking finding of how everyone at baseline is coming in already slowed. Reaction time. We don't know why that is. We can normalize that data so that everybody's at the same point, but it's very important to see this finding. Risha talked about ischemia and reperfusion injury. We hypothesize that this could be something similar going on in the brain from the repeated orthostatic intolerance insults that the body is taking from not recognizing post-exertional malaise, not recognizing that they have dysautonomia, orthostatic intolerance, so that, yeah, we have this issue with cognition. So again, a very sensitive assessment, objective and quantitative. This is speed and accuracy. Can't get much more objective than that for cognition. And very simple, 5 minutes. So, in summary, reaction time reflects how well the central nervous system is working, and it's a very sensitive measure to cognitive impairment. It's clearly affected by the orthostatic intolerance symptoms that are in both ME/CFS and in long COVID. We see very similar hemodynamic abnormalities in our ME/CFS patients and long COVID patients. And I mean, I think, again, the criteria, the IOM clinical diagnostic criteria, is a very beautiful set of criteria that can be used not only for patient care, but as outcome measures to really assess, objectively assess, the things that we are trying to impact in our various clinical research protocols and clinical trial protocols. With that, I'll take any questions.
Yes, thank you very much. There is room for some questions.
I have a question on the metric you showed. It was not actually reaction time, it was reaction time efficiency. Can you tell us a little about how it was derived?
So cognitive efficiency is an index that integrates both speed and accuracy. So that's a cognitive efficiency measure. You can even drill down more into the actual speed or the actual accuracy number. And the nice thing about, even though the simple reaction time is just a speed, because accuracy doesn't necessarily matter, or it does not matter in that particular test, it still is an output of cognitive efficiency in that particular formula.
Thanks.
Yes?
If you take the 400 millions with long COVID, and the most frequent symptom is a cognitive impairment, and some studies, or most of the studies, think that it's 10 to 20% have PEM. Does that mean that we have 20% of the 400 millions with new upcoming ME CFS patients?
Can I show my slide from yesterday? So we are about to publish a paper from the Large Recover Initiative study. Are you familiar with the NIH funded Recover study? That's looking at exactly that, to determine the prevalence and the incidence of post-COVID ME in the population of long COVID, in the population of infected. And broadly speaking, I'll give you some broad brush. Of the 12,000 infected people that we studied, 55% recovered uneventfully, 44% had long COVID, and then a subset of about 5% had post-COVID ME/CFS, met criteria for post-COVID ME/CFS. So we think from this work that post-COVID ME/CFS is a severe subset of the whole long COVID population. Did that answer your question?
Yeah. Thank you.
Susan, I have another question. Have you looked at other cognitive domains than processing speed? Memory, judgment, and so on?
We can, using Dana. It has other modules, but we did not, because it was really a matter of doing a very brief assessment from a diagnostic perspective, and again, from an outcome perspective, to just really zoom in on the reaction time.
Well, I don't think there are more questions. Now it's time for coffee. We meet again at 3 o'clock. Exactly.